You are seeing the screen, right? So let me take you to the next page. And as you said, we'll use NCBI. We will use ensemble. And let's hope that ensemble is working. Yesterday it was not working. And then you will use UCSC genome browser. Okay, so you will use a UCSC genome browser, and now we are going to UCSC genome browser. We are going to NCBI. Okay, so let me introduce you to the ensemble genome browser. So as the name suggests, the genome browser is something where we can browse information about genes, genome, and other things. So in the UCSC genome browser, we have many species you see here. Okay, so we can actually look for a gene, and you can also have different assemblies. You can have a look in human. So let me go to the human genome first. Okay, and then I'm saying I want to look for SERPIN I1, suppose. Any gene you can look. So now today looks like it will be available. Yesterday there was some problem. Okay, so what I see here, you will see a lot of information. So the name of the gene and description, what the gene is probably doing, what are the synonyms for the gene, and where the genome is located. So the gene is located on chromosome 14. And then you are seeing that the position, and it is in a reverse strand. So on a genome, you'll have a positive strand and negative strand. And we will see now. And so we are seeing about gene. So we are seeing about different transcript of the gene. And here you see that gene has different transcript. You're able to visualize. So tomorrow, if you are working in a lab or you have running your own lab, you want to work on one of the transcript, which is smaller than a usual transcript, then maybe this one. You just need to tap this one to know about. Okay. So this one thing you can do here, and then whichever transcript you need. But normally a transcript which has a full length. So human alpha antitrypsin is at 418 residual amino acid long. So this transcript looks like fine. This one also looks like fine. But one which has most of the information available in other databases. So this one we normally consider. So I'm just taking this transcript. Okay. So why I'm taking this transcript, I wanted to show you how the gene structure is. So exon intron structure you're seeing, then large non-coding UTR region, then a partially coding exon with the UTR region, then intron, then exon intron, and then again you have a full exon, which is a UTR, untranslated. Yeah. So untranslated is white. And so you're getting in this information. What if you want to see this information on a DNA level? That's also possible to see from ensemble. So if you want to see exon, you just say exon. This time I agree with terms and conditions to use it. Okay, so when I'm saying that 5' UTR region, then you have this first some exon is cutting, then you have this first coding exon. So and red regions are non-coding UTR regions. And then you have the first ATGs, one which gene has will start making methionine. Okay, for methionine, the gene protein will start, right? So first ATG, and then when let me zoom for you a little bit so that you can see easily. And these colors will come because of this lot of information is imbibed into it. You don't worry about those information at the moment. What you see here when the gene is exon is breaking into intron, you see that AG is there, and at intron, there is a GT is there. So AG, so you read in the books that intron breakdown, intron starts when the exon has AG and GT. At the point they break, there's AG and GT. So this is the first exon and intron junction. The second one also had AG and GT, and sometimes there will be some exceptions. So you see the exon and intron. So then you say, okay, no, I see that genomic DNA and cDNA. I want to see the protein sequence on map to it. That's also possible. You go to the cDNA. So in this, what Ensemble is providing us, these cDNA, the first line is genomic DNA, which is not coded. The first cDNA, the ATG, is producing methionine. 3:1 residues are mapped. Second amino acid is CC coming out of CCG, which is P, then S, 2 times S, and so on is going on. So as you see the full cDNA and what corresponding, this first line is genomic DNA, second line is cDNA, and third line is protein. So you can see, and suppose you are looking for one mutation somewhere here, you can actually map it very easily if you know it. So in cancer genomics, we will be again doing it to show you that cancer mutation maybe. So that's what exon intron you will get it because he is read in theory now. So you should also able to see in the browser, yes. So that's what you can see, and also you can see the protein sequence, obviously. The protein sequence of the gene, corresponding gene. Full, if you want to copy, you can copy from here. And intron junction are marked with the color. So this is splice junction. Other exon, so first exon, another exon, then another exon, and so on, is coming from different figure. Okay. So no, somebody is watching TV parallelly. So okay, so then this is what we can see, and that these similar things are visible in NCBI when you look SERPIN A1, and then I'm also going to put in UCSC genome browser. Again, multiple species are there. You can identify different species. I'm taking human assembly, and I'm again saying here also, sir, I'm looking for SERPIN A1. Okay, so I just gave both places. We will go back and forth because I do not want to go somewhere and finish everything another place you are. We have not started. So parallelly 3 years. So what 3 browsers help if you use more web browser? If one browser is making error, you can detect it if you are using multiple browser. So here in this context, again, all the transcripts are there. They have their own numbering, and you see the gene and transcript. And what you see here, you will also see the genomic locus. Okay, SERPIN A is there. Then we saw that multiple SERPINs are there in the neighborhood. These you can identify. And then this is you have to zoom left right to see that, but you can identify it. But in Ensemble, when you go, why use Ensemble? NCBI is not saying it's a genome browser. It's a normal browser. Normal, you can browse as database. So in Ensemble, you can see the entire picture of the chromosomal locus, right? So you see the SERPIN A1, SERPIN A2, all other SERPINs, neighborhood genes, all the classification you are seeing at the same time. And so if you are comparing this with human and also want to compare this with mouse, so what you just need to go to, again, do the SERPIN. We saw that there is a cluster of SERPIN, and as I told you yesterday, the A1 like SERPINs in mouse have much more extensive expansion. Yeah, so let me if we will reach here, we will able to see that. Okay, it's going to NCBI. Let me go back to ensemble. Another way is to going particularly to that species and looking for the gene. So I'm doing it another way. So I'm targeting mouse and I'm saying serpin A1. So this has recognized, but then again the same problem. Mouse. Okay, how can I reach there in this assembly of mouse? So you see there, it's easy to say, it's difficult to see. Sometimes you may have to tweak a little bit the software, and then I'm doing it. Serpin A10, A10. I'll tell you why. So okay. So this is a reference assembly. Should I get it? Okay. So I'll just tell you why I'm tweaking it. Okay. So now I see the same things. And then it is on chromosome. Serpin A is present in the same cluster where the serpin 10 is present. So now when we look all the serpins here, so you see that serpin 10, A10, all other serpin A, because serpin A in mouse has expanded, not just serpin serpin A1, but A1A, A1B, A1C. There are multiplied into multiple. So therefore, we could not identify one by simply naming serpin A1, because there are several duplications, and this leads into multiple serpin, more serpin than human has. So when you just chalk this out a little bit more, okay, so what you see here is that serpin are much more in number here compared to human full screen. So there are more number of serpin. If you count, the human has about 10, and then this one has about. So this is the starting one: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. Still going on. So 21. I will at least 30 of them. Yeah. So that's what mouse has the expansion. So expansion in the same genomic region. And so you saw that one can detect it. So if we go to a lower species, the number will decrease, because we already have seen the hint was there. So when you go to a fish, number will decrease. So one can practice by decreasing number. That will be much easier to detect. And that again, we can do that maybe to have fun. And okay, let me go to change the browser. I'm going to CSC genome browser again and looking for human serpin. Okay, so I saw that there is a human serpin A1, and I want to zoom it for 100 times and see what happens. So basically, these are blocks. We can actually take picture of it, right, and keep it in a PPT and take other species block and take a picture of it, and we can see. So you see that this is A1 of human, and maybe I make it even smaller. And in this one, you can zoom in and zoom out. And I said 100, so it's like zooming it 100 times in, so therefore it's taking time. And also everything is happening in the browser, so browser will lose whatever has to be lost, it will happen in browser. Yeah, so now what I see the lot of genes, almost many things. So let me zoom in at least 10 times, so 100 times twice I did, and now I'm reverting back because there are a lot of genes here. So still 3 more times I'm doing. Okay, so now what we see here is that lot of genes we are seeing, and we were focusing on serpin I1. So it's Serpin I1 has moved here or there somewhere because of jumping. So I'm just writing it again. Serpin I1 I'm looking for, and then I can say I'm looking for okay, so it's giving me back. Okay, so I just can zoom in 10 times and then mm, 10 more times. Okay, so what we see here is that Serpin 1, 10, 12, 4, 5, 3, all are available, and these are different times they are repeated because they have different transcripts, and then their cluster other neighborhood genes are there. And in this browser it looks different, and you can just take a screenshot of it and you do it for mouse and a screenshot of it, and so we'll see what are the difference between them. I do not touch it actually. So in this browser, you can actually explore a lot of information from different sources. And these are here, what provided here: mapping and sequencing, gene prediction. So if you click on, these are called tracks. And you can click on, then you will start getting information about these tracks. So for example, this one has with OAM allele frequency here. It does also have gene expression data for each gene is available here in different tissues, and the colors are showing you different colors are different tissues actually. So the expression pattern is shown here. Then it is also showing in how conserved this region is between the human and human, mouse, rat. Human we have on top, mouse, rat, dog, elephant, uh, zebrafish, frog, and all. So this can be tweaked on and off. So basically, if I'm not looking for this, suppose of expression, yeah. So this one is shown here. GTX is packed. Now when I hide it and see what happens. So one can change it what you want to look. So the earlier bars are missing now. So this is hidden. Suppose this encode non-coding data is also available. If I go here and encode, I can close it. I can just hide it. This one also I can hide it, and I just refresh it. It will be gone. So it can actually invite a lot of information for the genomic region from this, and now it's hidden. So one can hide and play on and off. And obviously for human, there will be a lot of data available. For other species, less data will be available because human is the top reference genome. And so you get a cluster both in these browsers, but in a slightly different way. And then you have to compare. In a comparative genomics, you will compare it. You will make use your manual inspection, other way wise, programmatically, try to figure it out what you get. What we have seen so far is that mouse to rat has mouse to human mouse has more number than human, but okay, so ensemble can alone also fulfill some questions, so that's not a problem. Suppose I just want to go back to ensemble and I want to see what is the state of Serpin A1 in the species. You can check in any species. So I am just checking into zebrafish, which is done your area. And that says that gene is available on chromosome 20. So now it's giving you the list. And then what you expect this to have. You have already seen the theory. So we expect 1 or 2 genes in the cluster. And that's what we see here. Okay, so there are only 2 genes in the cluster. Right, so that's what we expected, and that's what is available. And so fish to human, there is expansion, and expansion is even greater in case of rodents, and that's what we saw. And when I'm saying rodents, so we can go here and run. Say again, same thing with we can do it for not for zebrafish, but for when you are saying this rodent, can we see this in happening in rats? Mouse we saw, rat. So rat. Okay, rat will be near rodents, right? So rodents are placed together. Yeah, rat. Huh? So again, I say serpin 1A1A because there's this nomenclature they have A1A or so A1A is not there. So let me see. So am I looking into okay in rat? Okay, serpin A1 is there. Let's see. And then it's in chromosome 14 and then lot it's human one. It went out and gave me a result for human. No, we're not looking for human. Yeah, I'm looking for Norwegian rat. Okay, rat gene. Okay, this is in chromosome 6, and which our objective is to just see if the cluster is multiple or not. Yeah, so we see that more number of serpin are available here also, and we can go left and right to confirm it actually. And some of them will have a locust-like name. So we see there are a lot of serpin who have which are more than human, but many of them are maybe called locusts. So we have to see whether the annotation is proper. So I'm not going very distant. I'm going from human to mouse and rodent. You see that annotation within the top reference genome also is a problem. So one need to be carefully watch for the names and look for the names. Yeah, so then you can also practice. You need to just practice, or you need to build a project. If you are interested, you can contact us. And then we are going to see what all we can actually do. So this is ensemble. Then you wanted to do this. Yes, we'll do that. But okay, so some of these things we'll try. And obviously you can get the tree, do the analysis, and get the map. Then if you construct multiple map, it's called synteny. Yeah, multiple map the way we have made synteny builder ensemble. So I think there is a builder which is available. Yes, in portal there is a free app where we can build it. But the app will not give you what you want to see. The synteny the synteny I do not want to use this. There is an inbuilt in the app for SynDB. Let me see. So there are different tools you can also use, but then your data is available or not. So synteny builder there is one synteny builder in ensemble also. Let me see. There is one here in ensemble, but then it will build in entire chromosome. So who can't see what is happening. Okay, so then okay. So you can compare with human chromosome and the chromosome here. So there are a few tools for building that automatic figure, but the automatic figures will be not very informative. And now I just want to show you that in this gene here, these are the contigs where there's a gap here. So this region is missing. This is in rat itself. So you see that some region is missing. and that's what we expect to have. Okay, so yeah, so this is what we can see there within a not and rat is one of the uh, standard organisms, so we expected it to be full, but the errors are always there, and you have to make it. And when you look at, if I'm saying error, so it is just in a big intron, so that it may not be have a problem. But normally, if it is in the coding region, suppose here, you will see partial sequences because of this. So now let me build the anti-cancer drug. So these are clusters. So you are always counting on the cluster, and then we are looking for anti-smash cluster. So anti-smash, we can have a look. So the NTS-MAS has, you can load your sequence from NCBI, or you load your local sequence. You take your sequence and load it. And then you would need to give your job. So the jobs will be running, and it will give you result. So load an example. So just I have loaded an example. So you loaded, you need to give email ID and you can run it. Okay. So then you run it, you can open these different parameters, and it will run and give you result maybe tomorrow, day after tomorrow. There's a bacterial, there's a fungal, there's a plant, but fungal is not working at the moment. So if you look at here, there's a problem. Server moved to outrest. So the bacterial one is using. Let me have an example of here. So what this will tell you, that these are the different clusters. So if you look in the cluster, this one is a terpene cluster, the second one. So mark in a different color. So again, if you look second color, terpene clusters are there. And then you have the hybrid cluster, which has light green. And this is cluster number 1, or region number 1. So what you see is that there's gene cluster here, and corresponding proteins which are forming the cluster, the core member of the cluster. So red ones are the core member genes, and then these are the protein domains on them. And then you can also get, suppose I click here, you will also get information about it. And then you can also connect to NCBI for blasting. So if you can to know more about the gene, you can blast in NCBI. And then if you know more about, so if there is a drug-like molecule available, it will be also be here. Structure is available, or you have to pick it with other tools. So and if this cluster is formed in other, so this is streptomyces cluster, and then you will find a lot of us streptomyces have this cluster. If the cluster is known, obviously it's easy for you to pick the drug, because maybe some of the streptomyces has known. And this known cluster blast, yes, so this is having a known cluster blast, so it's known in, yeah, so this is potentially producing linomycin. Okay, potentially, yeah. So one need to do a lot of chemistry about it. Okay, so this is true that streptomyces are producing this cluster. So this is the compound. Again, similar figure what you saw from so biosynthetic gene. So it can happen that this cluster is indeed producing this. You have some knowledge now, and then you need to compute these domains, whether these domains are essential for production of linomycin, and so on. And then, but this is clearly a hint that it is potential producer of that cluster, because even a species is matching. And then in the cluster, as I said, we will have different accessory genes. So let me see there what accessory gene is. Okay, so this is additional genes, transporter genes, regulatory genes, all other gene resistant, ATC domains. So a lot of information is already available in this cluster. When you go to next, once you completely analyze one cluster, you can analyze 10 second cluster. Second cluster has something, and then second cluster is also found in different streptomyces. Let me see if it is known. Now it says that it is producing a carotenoid-like compound, and maybe rustimycin. We don't know. There's some hints are there, and then one needs to be carefully check whether. So in some subcluster is not known. If the gene is, I'm clicking on the gene. If the gene has information, if there's a drug-like structure is known, that is also visible here. So let me see. It's not, they're not much known. Maybe I take one more example of NRPS. Okay, now you see the different protein domains here. Different gene is there. Maybe I take one of this, and now you see that different subunits are, so subclass protein. This gene will have different units, and these units are shown here. And now potentially this is showing us that what kind of amino acid is produced from this cluster. So it's giving hint about what kind of amino acid is produced from there, so the circularization is possible or not. And so on, you will get 50 cluster. You need to analyze 50 cluster, but you will get an idea of the cluster, and you already have your genome, so you can match with the cluster and get information. And by doing so, you can deduce knowledge about future drug-like compound. And it is, I'm saying it, it takes time, and so it's one of, in our lab, one of the two masters BTech students have worked 6-8 months. They analyzed lichens for producing such drug-like compounds, and they are writing their paper. So it takes time. It's not. It's easy for me to tell you, look here, look here, look here, and one need to pinpoint what is happening here. Why there's so many genes are required in the cluster, what they are doing. So these all need to be get hint of it, and it's not easy. It required time, and so on. So basically, and it's a combinatorial. So this large cluster requires together like operon, and then in the bacteria definitely it is operon, but in eukaryote also it serves as operon. So that's why, and that's not just produced by bacteria, plant, fungi, some algae also produce secondary metabolites like from the clusters, from the gene within the genome. So whole idea is that use comparative genomics to resolve genome to drug problem. Yeah, so from genome you could make prediction of drug. Okay, so this this is all we wanted to practice. And suppose you want to do a gene structure prediction, you need to go to NT-SMASH, not say Augustus. Yeah, gene structure Augustus gene structure. Yeah? So let me have a look here. And so we can go to the web interface, web Augustus. There is a web interface also. This is in the University of Greifswald currently. And then we can do the prediction. And you can do, there are a lot of things you can do, make submit prediction. Maybe you just need to submit an example, fill in with the sample data set. So you need to provide some example, and then you fill the form as they require, and then a couple of days later they will send you the result. And if you are computationally you are able to do it prediction yourself, you need to just download Augustus from the web browser or GitHub. They have it now in GitHub, and then you can actually download it and use it with the in your Linux machine. It will work, and you need some training. So if you want to run, you need to give the file, and they have given. Okay, so prediction tutorial is available. So one can go here and run. So you need to fill the prediction summary. Then you give an email ID. Obviously, you need to give email ID and species parameter. So you have to give a kind of which species it is. So kind of suppose your species has detailed information about archive, you can give, or you can choose out of the list which is the closest species uh, of this the project you are. Suppose you are running for a fungi, you will give a related fungi, say Aspergillus origi you will give. So it will give you an you are providing an hint is the closest species. So gene for human will be different than gene for fungi, because the intron exons will be smaller compared to human, because human has a lot of repeats, fungi will not have. So therefore you need to provide a hint to the algorithm that what kind of, and then you have a chromosome will be looking like this. So this is what you are filling it. So this pasta file will be like this, and chromosome 1, chromosome or scaffold 1, 2, 3, and then you need to upload your data maybe from Excel, maybe in a text file you have it, and then you need to provide, if you have CDNA information, it's optional. If you have CDNA information, which primarily not found in many cases, no need to worry about, it's optional. And then it will give you, you can provide as many as data you want, but it can have, if you give give one example maybe here. Okay, let us go to ensemble. No, NCBI itself. So we know. Okay, so let me take you some. You just need to give it DNA fragment. So let me have Aspergillus genome. Maybe one genome you need Aspergillus. I'm making error. Google will correct it. No. So Aspergillus. Okay, so there's correction to level. Okay, so Aspergillus is there. So what I need is one of the nucleotide I need. Yes, just we need a nucleotide, not full genome. And just to train you a little bit. And I am seeing that there is a scaffold 46 just we're getting from some species. Okay, so we just need a faster sequence. Okay, so faster is coming. So you see that penicillium is scaffold 46, and this is a big file, right? And then you need to so in any chromosomal a random genome will look like this. Notepad plus plus. So it will be, say, it has name scaffold. You can name it anything you like, say chromosome 1, and then maybe I have written 2, right? So let me write 1, and then if you want to copy it and repeat it, So then it becomes second chromosome. So I mean, obviously DNA will be different, but in a random scenario, you can just have two of them. I'm giving you an example of how the chromosome, how the genomic file will look like. It has nothing. So that's what it is called. That's what you need to annotate and predict genes and everything you want to do it. So I'm just taking it and putting in a desktop. For, so I'm saying a random genome, yeah. Random genome. Suppose you have in hand your random genome, which you don't know what is inside it, and it has two chromosomes as we named it. Now we have two chromosomes. We want to make prediction. And let me see if the easiest way of making prediction. Uh, okay, so we go to the Augustus, and it's at one point we came out here. Maybe there they have web interface. Yeah, this one. Okay, so there I just need to on web page itself we can give copy and paste, and then this is older version of it, but it's running I think on the web page itself. So we know that we given some aspergillus or something, just one closest species we have to give, assuming that we don't know which species it was. And then we say, okay, both a strand, yes, we'll run on both a strand, look for gene alternate. You can say a few alternative splicing and run it and see what happens. Okay, so this is calculating, yeah. So if you are running a local on a local server, or if you have a Linux installed where the Augustus is installed, this is what command will look like. So they are using it on their server, data www on their web server they are running, and the Augustus bin has Augustus program, and then a species which this whatever parameter we have given that it has taken in. So if you know these parameters, actually you can also run by installing locally. I'm not sure how many of you are using Linux or Windows terminal. You can install the software on any computer which is having Linux like or Power PC also, Power Cell also, in Windows also. So in this computer we have it actually. If I look for the cell Windows terminal, so I assume that many of you know that Windows is using terminal nowadays. They use the Windows Linux cell inbuilt in it. You need to optimize. So going to use Ubuntu. Now it starts as Ubuntu 20 without going in, without doing anything. So I'm not doing opening for you a VirtualBox. It runs on Linux. So it's you see that some files are there. So it's running Linux. And so this is the command. If I have, I can copy it and modify a bit and run it in my Linux. So assuming that at the moment it is still running, so the result will come graphical as it says. So graphical result, browsable result. So I'm just taking one example if it is available. Okay, so this is how the result will look like on graphical interface. And so this is how an output will look like. There's nothing here, so it's not showing yet, but it's still calculating. And these are the comment lines. So this is that Augustus is running with this parameter. And what will be output is still not output, not generated. So it's running, and it will give given a result. So as you see, online also one can run, at least train. And obviously when you're doing, you need to optimize yourself, optimize with the data set, and do the gene prediction. So this is one method of making gene prediction. And so you can say, okay, can I make a gene prediction with eyes? Yes, it is possible if your eyes are smart. If you have one or few genes, of course you can do it. So suppose I have this any gene from here, we can take it, and if we have no start codon and next codon and third codon, 3-4 codons, you can pick from there the gene starts and start coding. Randomly you can pick it, but yeah, it's more than one gene, then it's not advisable. So there was another tool earlier used to be available. I am not sure whether it's available now. Softberry, where you only need to Softberry gene prediction. Still available, but let's see if it is allowing us for free and run online. Okay. So Softberry can run some programs, and okay, let me see if it is still running. Okay, this is running, but no, not many people use it. This is 2006 program in front of Augustus. Very few people use it or not use it. So I am pulling my DNA here and I'm giving it. So it is also asking a hint. The reason is very simple, because gene structure and intron length and all very different, very different in different species. So therefore, you need to provide hint to the program. So the no universal law will be followed. So ascomycete it is okay. I'm giving this because our sequence was ascomycete. So it will try to predict something. There is error in nucleotide. Okay, so maybe they just need one, not two parameters. So let me have. I'm just randomly fusing the positions. So let me try to run it. If it runs, error. Okay, let me go back. So it is asking for some change. Let me give this one. I've taken a chunk of DNA here, so you don't know whether gene is there or not there. But program will try to pick whatever it can. So yeah, so initially before Augustus came, we all were using this, and I'm not making any guess. Let's see. Error in reading nucleotide sequence. Okay, so it's saying there's still an error. Oh, this one line here. Okay, I'm asking it to. Okay, so there's a no reliable prediction. Why don't we just go back here and give it a full pasta text. I will just give the full length. Okay, so let me give it full. Whole idea is to give you a visualization of gene prediction. Meanwhile, we'll check this calculation is still going on. Okay, so 5.5 MB. It's a big size for any online experiment. So it's a one scaffold equivalent to one chromosome. Let me see. We just need to maybe drop. Let's see if it's working. Search. So program itself probably went out of the program itself has less memory. Okay, let me So one can export from here also the genomic DNA for this region. So just exporting a random location from mouse genome. So feature, FASTA sequence, bed sequence. Okay, I'm just trying to get some sequence for our prediction. Okay, so this is not bad. This is not bad. This is not very big. So this can intron protein coding. So I'm just looking for chromosome 6. I'm still trying to see if okay, this one is looks like genomic. So if I give it to Softberry, I'm giving a mouse one, a rat one. So let me see if a rat is available. Ratus. Kangaroo rat is there. So you see that mammals are many of the mammal are available. So ratus is not available. So let's see, the moose is there. Mouse generic. Okay, I'm just checking mouse. Closely related species one has to give. And I'm running. Okay. So it made a prediction. Okay, it says that 4 exons are there, ATG and HEG. So this is very simple task I give to run, and then it shows that there is a 4 exons. It is starting with this mRNA 4 exons, and this is the this is the so basically if you compute this, you will also get your codons one by one, cDNA, and so you can make a small gene prediction from a study. But if you have to make 20,000, obviously you will not make 20,000 times a different permutation combination. So this is, and then you can get the picture in this one. So it's still working. So I think I used it last time 2003 also. So you see that the CDS distribution and result is available here in nice pictorial way. So one can practice also, and one needs to, if you want to do comparative genomics, you need to figure it out what you want to do and why you want to do, and then how you'll proceed step by step. And if you proceed step by step, the next step will be clear. If you want to conquer anything, you have to break into pieces. So with this, I hope you have learned, and I have given you good examples. And I'm open